Okay, we'll get started. Thank you everyone for joining us. Today's webinar is Asiatic Garden Beetles as Emerging Pest of Corn. My name is Laura Isles. I'm one of the co-directors of the North Central Integrated Pest Management Center. We're one of 4 regional IPM centers supported by USDA NIFA Crop Protection and Pest Management Regional Coordination Program. We have a mission to coordinate IPM across the region by sharing and promoting IPM research and extension. If during today's webinar you have any questions, there's a Q&A portion at the bottom of your screen, and after the presentation we will answer all questions. So I'm really excited to welcome Dr. Kelley Tilmon from The Ohio State University to share with us all about Asiatic garden beetles. I don't know very much about them, so I will let you take it away, Kelley, and introduce anything more about yourself and then the project.
Thanks, Laura. Appreciate the introduction, and thank you everybody for being here today on what's probably your lunch break. This is a story about a new field crop pest that we have in the North Central region, in particular in the Great Lakes region. And this is Asiatic Garden Beetle, a scarab grub. And I have put as first author on this talk Adrian Pekarczyk, who was the graduate student on this project. And so he actually had the good fortune to get a real job right out of school when he graduated, and he's now a research scientist at the USDA ARS lab in Brookings, South Dakota. And the other collaborator on this project is Dr. Chris Stefanzo, a field crop entomologist at Michigan State University. She is my Michigan counterpart to the field crop entomology that I do in Ohio. So this story begins in the middle aughts when something weird started to happen. And typically in this region, we have not had early season pests of field corn. But in northern Indiana, northwest Ohio, and southern Michigan, field crop entomologists started to see significant plant stand loss. And this was something common to these regions in all 3 of our states. This is another field with stand loss in southwest Michigan and northern Indiana. The culprit was identified as Asiatic garden beetle. This is a scarab. It's an annual white grub species. But the thing that is really strange is that it had never really appeared as a field crop pest before. This was a new thing. Corn is the hardest hit crop, but we also occasionally see damage in soybean, alfalfa, wheat. This is damage that happens from the grubs feeding on the roots early in the season. And also meaningfully, potato industry in southwest Michigan has been hit by this white grub. You can see they feed on the tubers rather than the filamentous roots. And you can see a little grub peeking out of this hole in a potato. And Chris Stefanzo has estimated that in 2016 there was about $1 million of loss to the potato industry. Historically, we have only known this pest to be a very occasional and fairly minor pest of turf as a grub, or also occasionally as an adult feeding on foliage, a pest of ornamentals, and not really hort crops so much, more ornamentals. So what was going on? Why were we having yield losses up to 40% in Midwestern corn in the Great Lakes region? Well, we had a problem on our hands because of the history of this insect. We knew virtually nothing about it as a pest outside of turf or ornamentals, or for that matter, outside of the East Coast, where most of the research a long time ago had been done on this insect. And so we submitted a proposal to the USDA Crop Protection and Pest Management Program, subprogram Applied Research and Development. And on our second try, we got a grant to work on this problem in the Great Lakes region. So a little bit of history about where we started with this insect. This is an introduced insect from Asia. It was first discovered about 100 years ago in New Jersey, where that yellow star is. And by 1935, it had spread along the coastal US, mainly in the Middle Atlantic region, and made a name for itself as a pest adult in a beetle pest of ornamentals, vegetables, and small fruit. Almost all of the research available on this insect dates to the 1930s, from this time period where it was a foliar pest as an adult in the Middle Atlantic region. And most of this research was done by a man named H.C. Halleck working for the USDA. Gradually, it spread beyond the East Coast, mainly known as an occasional minor turf pest. This was the distribution in turf, at least up through 2000. And it was never really much of a pest. It was sort of something they found along with other white grubs that were causing problems. This is the known distribution of Asiatic garden beetle as of 2012, so really a very broad distribution, but again, not known anywhere in this range as a field crop pest until this time period, and in this particular area starting in the middle aughts of this century. And this is an example of the type of stand loss and stunting that we see as a result of the grubs feeding on the roots. So in this picture, we've managed to capture the root profile of this seedling corn plant. You can see the little grub down here and a blown up image of what a third instar grub looks like. So not only do we see stand loss in terms of plants that just die, but we also see a lot of stunted plants. And we've seen yield losses up to 40% of the field as a result of this pest. We have indirect yield loss from late replanting. We have costs that occur from replanting. Because this is a soil-borne insect, we don't have rescue treatments. But this was a big problem for growers, no doubt. They definitely were noticing. These are some of the quotes from both growers and crop consultants that indicate the level of their distress and just how widely it was being found. One of our crop consultants, the bottom quote, "I am finding the AGB throughout the tri-state area. I am amazed at the populations I have found." Incidentally, for any early career scientists who are contemplating grant proposals to NEFA, it is really helpful, I have found, to include letters of support from the stakeholders of interest, as many as you can get your hands on, even if they're not required. It's a great idea to put it in. I think reviewers respond very positively to these letters, and we had almost 20 letters of support from farmers and crop consultants to do research on this insect in our region. And one of them was even a note a farmer had scrawled on the back of a receipt, and we took a picture of that and included it along with the others. So what did we know when we were going into this about the biology? Where were we starting with this insect? Well, okay, so we knew it was a scarab and we knew what it looked like and how big it was relative to other scarabs that are occasionally found in field crops. This is the Asiatic garden beetle here compared to it's a smaller than some of the others. It's about a quarter of an inch long as an adult. Fortunately, the grubs, white grub ID can be tricky, but this species has a very nice character as a grub that allows you to identify it easily, and that is this sort of a pouch structure on the side of the head. They also behaviorally are a lot more active than other grubs, white grubs that you find in field crops. They really squirm a lot. They'll even bite you, which is not typical grub behavior. But this cheek pouch, as we call it, is a good defining character. This is an egg, and this is a little first instar, and this is a third. This is a third instar. From the 1930s work, we know that the adults have a very broad host range of over 100 plant species. They attack just really anything from fruits to vegetables, ornamentals, weeds, native or non-native, doesn't matter, woody or herbaceous, doesn't matter. Very broad host range as an adult feeding on foliage. We know that the adults are nocturnal and that they are attracted to light. We knew this from our own observations of finding them on the sides of buildings at night. In agricultural habitats in our area, we could find the adults feeding on weeds in the field crop area. Very seldom do we find it feeding on the crop plant, the adult foliar feeding. Mainly we find the feeding in the field as adults happening on weeds and the broad range of weeds. They really like mare's tail, but they have plenty of other plants that they will strip down to the nubbins. And they also, we know that the adults they're nocturnal, and during the day they'll kind of snuggle down into the soil underneath their feeding areas. So these little orange things are Asiatic garden beetles hiding under a mare's tail plant during the day. Just a few more weed pictures, ragweed, Queen Anne's lace. Anecdotally, we knew that the biggest problems, the hardest hit fields, were fields that were very sandy. That is not the case in turf. When you do find it in turf, there's no real correlation that people working in the turf industry have observed with sandy soil. And in fact, some of your most famous turf habitats are not even grown in sandy soil, like, a lot of golf courses. So, not a lot to go on. Where do we begin? And it was kind of exciting to be working in a system where you really are kind of starting at the beginning. Well, first, we needed to develop sampling methodologies, in corn in particular, our focus crop. We needed to establish life history parameters in our area and in the crops where we were interested. We wanted to take a first stab at management because not a lot of things seemed to be working. And then we also wanted to tackle other interesting questions that came up along the way. So if you can't sample something, you can't study it. So that was job one. How do we sample these things in field crops? So sampling the grubs was the first thing we tackled. And there's different white grub sampling methods. Um, one or another of these might be better depending on the habitat and the insect in question. There's something called a compact cutter, which is designed to take a shallower but broader soil sample. There's the golf hole cup cutter, which takes a core sample that is deeper and narrower but deeper. And then, oops, looks like my bait station image is not showing up. Anyway, just spoiler alert, bait stations where you have a strip of, it's a metal strip with sort of loops in it, and you plant your little plants that are the bait and see what comes to it. Ineffective and a real pain. So between these two, cup cutter and compact cutter methods, which works better? Well, we found that after adjusting for the soil volume, we get significantly more Asiatic garden beetle grubs with the cup cutter than with the compact cutter. And the reason for this is because it takes a much deeper sample in the soil profile, and these grubs tend to like to go a little deeper than some other grub species. How do we sample the adults? Well, some of the go-tos that one might use, the classic sticky card mounted above the vegetation. And because these things harbor on the soil underneath debris and plant matter during the day, when they come out in the evening, they're going to be crawling around on the soil, maybe a pitfall trap. Jumping to our conclusion on this, pitfall traps win by a mile. We basically can't even find them on sticky traps. I think in this whole experiment we found one on a sticky trap, but we find plenty in the pitfalls. So pitfalls are really the best way to go for sampling adults, especially in a small plot or moderate plot sized setting. We also looked into adult sampling that anecdotally we got a lead on because we were doing monitoring for Western bean cutworms with these very simple milk jug traps with a Western bean cutworm lure and antifreeze, the dog-friendly type of antifreeze. And anecdotally, when we were checking these traps from Oz, we found bucket loads of Asiatic garden beetle. And so we were wondering, can this be used as a sampling method for flying adults? And what we found is, yeah, sure, we can catch the beetles this way, but the resolution is very low, by which I mean, like every trap in a township might have a uniform collection of Asiatic garden beetles. It's not something that you can use to detect the sort of plot by plot variation that you would need to know about if you were actually performing an experiment. So it might be useful for kind of regional sampling, regional populations, but not for experimental work. And there are always hazards with research, and we encountered certain sampling hazards. So in due diligence to using exactly the same type of antifreeze that was in these milk jugs, we looked for the pink antifreeze from Meyer grocery stores because it was Meyer brand uh, antifreeze that one of our county agents had first made this discovery with. And so we went to Meyer. The closest one to Worcester is, I don't know, like 30 minutes away, and bought out everything in their little automotive section, which led to a nasty gram from OSU purchasing. I'm sure all of you who work in this area have gotten similar nasty grams wanting to know exactly why we were buying antifreeze and how this related to our work. Also, amusingly, at this Meyer, we went to the shelf area and took the antifreeze from there. And then when we went back a couple weeks later, we noticed that they had restocked with an actual bump out on the end of the aisle with this antifreeze. They had clearly decided there was some major pressing need for antifreeze in the area, and they were going to take advantage of it. Another method we tried to trap adults, we know they like to come to lights, and so we bought a bunch of these tiny little battery-powered LED lights and made holes in Rubbermaid tubs and to make sort of there's a hole here to try to make sort of a baffle trap for flying adults. This had a very unexpected hazard of, yes, raccoons. Chris DeFonso was the first one to beta test this trapping in a field in Michigan. And the night after she put them out for the first time, she wrote this to me. Raccoons like pitfalls and pulled a few up to check them out. Raccoons love tiny lights. So fascinated by them, they plucked every light out from the tops of the cages except one, n equals 17. I found no AGBs in the cages, but maybe the beetles flew out of the hole. I also believe the raccoons stuck their tiny dirty paws in the cups of antifreeze. There must have been a whole tiny masked gang of raccoons rustling through the field to mess with all of my stuff like that. They found every single one. Raccoons felt the need to pee on some of the traps and left me a tubular fecal present in one pitfall. Thanks. So apart from the fact that raccoons were a hazard, this also didn't turn out to be a very good sampling method. So we moved on. Okay, so now armed with proper sampling methods that we could effectively use, we next tackled life history. That's before you do anything with management, you really need to understand the life history of an organism. The first thing we did was some morphological work to be able to separate males from females, and we found a new character that hadn't been described for this insect before. It's a ventral abdominal character, and this work is published now. And then we moved on to looking at the life history parameters in the Great Lakes area. And this involved sampling in AGB infested fields, 16 of them in both northern Ohio and southern Michigan. And in each of these 16 fields, we visited them weekly for a time period stretching from sometime in April to early August. And in each field, we established a 5-acre grid. We sampled for grubs, what number of grubs, how big, what instars were we finding when. We sampled adult beetles, their numbers and timing. We sampled for each grid, for each cube in the grid, I should say, for each slot within the grid, we sampled soil type and assessed the amount of weed coverage. And that allowed us to come up with some very detailed life history, life table type stuff. There are 3 instars of grubs, first, second, third. Um, there's a lot going on in this graph. For communication purposes, we reduced it to a more user-friendly graphic where we can describe what's going on with the life history. Grubs, they overwinter as grubs, it's mostly second and third instars. Starting towards the end of March and in through April, the grubs that overwintered start moving up higher in the soil profile. They go down deep when it's cold. As the soil warms, they start coming up. They are hungry, they are ready to eat, and this period where the grubs are moving up also happens to correspond with the prime corn planting time in our area, which is why we see such problems at this time of year. The grubs are coming up, second and third instars, they're hungry, and here are all these nice little corn roots ready for them. After the grubs pupate, which happens in June-ish, they start to emerge as adults in in parts of June and also into July. The adults mate, they lay eggs in the soil in late July, early August. In the late summer season and in the early fall season, these eggs hatch. First star instars begin to feed. First instars never causes a problem, they're too small. As the soil cools, they work their way back downward into the soil profile. By this time, they're second or third instars, and they go into their overwintering. The Great Lakes area has a very complex geological history associated with strange patterns of glaciation. And the upshot of this is that we have a lot of sandy soils in this area, where also we're finding the AGBs. And also we have tremendous soil variability. Even within a field, you can have really different types of soil, even in the same field, because of how the glaciation deposited things. This is an aerial view of Asiatic garden beetle damage. And I mentioned we knew about the sand. This was from growers. If you asked a grower, where are you having your AGB? This is a quote I love. They're up on that sandy knoll over there. I want to draw your attention to these fingers of damage right here. And this is interesting because this is a soil map of that exact same field. And look at these fingers of loamy sand. Matches pretty much exactly to those fingers of damage. Whereas beyond those fingers, we have a different type of sand. There's a difference between sandy loam and loamy sand, I have learned in the course of this project. This type being much sandier than the other. So we knew this anecdotally. It was never really a question, but as scientists, we have to document, right? We have to get the numbers behind it. So when we were doing this grid experiment, we also took sand type from each grid, and sure enough, we learned that Asiatic garden beetle infestations begin at about 50% sand content, and the grub numbers increase linearly with increasing sand. And this is when you adjust for all other variables like, location in the field and when you were sampling and all of that, we still see a very significantly greater uh, proportion of grubs in sandy soil. Now, remember that I said that the adults feed on weeds. Well, we also wondered, does that uh, result in a correlation in the field with weedier areas having more grubs? So also in all of these grids, we took percent coverage by weeds. And surprisingly, we were surprised to find that we did not see a correlation, significant correlation between grubs and the weediness of the patches. It was more or less the same. Okay, learned a lot about the life history. Next stage was to try to tackle some management. Not a lot seems to work on this grub. We knew some things that don't work. Surprisingly, neonicotinoid seed treatments, things like Cruiser, for example, or Poncho, don't really seem to work at all at low or moderate treatment rates. At the highest possible seed treatment rate, even that doesn't really work particularly well if the if the garden beetle pressure is high. The most common in-furrow products that farmers would use at planting do not really seem to do anything, and nor does tillage. You might think breaking up the soil, tossing things around might have an effect, but at the very best, we might see only a minor suppression with tillage in the experiment we ran. So what other management options might we try? Well, okay, there are some less utilized in-furrow chemistries that don't get used very often. Maybe something in there would work. And then we were also interested in biological control, in particular entomopathogenic nematodes. We did a bunch of LD50 testing in the lab for different in-furrow products. We found that products that contain only bifenthrin, a type of pyrethroid, for example, Capture is a very popular insecticide used in furrow, do not work at all. And I mean, they do not work. At any concentration we tried, we couldn't get the upper limit of the upper limit of toxicity because there was no upper limit. I think the highest we tried was 4 times the label rate. And I think you could just open up the bottle directly from the dealer and dump it on them and it wouldn't kill them. This is important because bifenthrin in-furrow products are really the go-to, at least in our area, the go-to for most farmers and crop consultants. In fact, when I was presenting some of this research at a pesticide applicator training event, when I finished, one of the crop consultants stood up and he was like visibly upset. He said, I was telling my guys to use Capture. I was telling them to use something that doesn't work at all. Like he was upset that he had been, and the best I could tell him was, well, that's why you come to these training events, and now you know. So this is why we do the research, and this is why you come to hear it. What did work was something not used very often, chlorothoxophos, which is an old school organophosphate. And we found that to be very effective. Now, only one chemical company in the US sells this, Amthac. The liquid version is Index. The granular version is called Smart Choice. It's mixed with bifenthrin, but I don't believe the bifenthrin is helping. And both of these products work very well at not only the lowest labeled rate, but even much lower than that. Like I think, as opposed to Capture, you could just take the bottle of this and just sort of wave it over the field and you'd get mortality. Drawback, however, this, as I said, is an old school organophosphate. These are very toxic compounds, including hazards to human and applicators. In fact, this can only be applied with a closed delivery system. In the case of the liquid, it's an injection system such as Dosatron or Raven. In the case of the granule, it's something like a Smart Box, which is a closed handling system. where the applicator never has an opportunity to come in contact with the compound. And so not all farmers, in fact, not even most farmers, have this equipment. So it really limits who can be able to use it. We also wanted to do field trials, but I will say that field trials are very difficult to line up because you are really, in our case, we don't have this pest on any of our research farms, and so you're restricted to on-farm research, and that means you're restricted to, first of all, finding a grower who's willing to work with you on a very inconvenient planting scheme. And you're also restricted to what the farmer is already set up to use and willing to use. That said, we were able to find farmers who were willing to do strip trials with us of uh, two um, two products, one product at each location, and those were Aztec and Counter, which are both organophosphates. And because the organophosphate that we tested in the lab was very effective, we had high hopes that this would be effective. But it was not to be. This is the graph showing the results for Aztec, black bar, untreated control, gray bar. No difference whatsoever in beetles. And it was the same result for Counter, which I'm not going to show. So that was disappointing, but we turned our attention to biological control. In particular, my grad student Adrian was very interested in entomopathogenic nematodes and their potential to control soil-dwelling insects. So the first step was figuring out what's already out there that we can isolate and rear. And he was particularly interested in um, what can be done with existing nematodes rather than adding, new nematodes to the system. And so the next step after figuring out and isolating and rearing is lab testing them for how well they kill Asiatic garden beetle. So this involved soil field sampling at 10 different locations in northern Ohio. So go out, grab the soil, bring it back to the lab, and then there's a protocol that you can use to bait for these nematodes with waxworms. And then you isolate the nematodes in water with this thing, sort of this plaster coated white trap. And by doing this, Adrian found 4 different species of nematodes from our sampling. One of these was problematic to work with, so we did greenhouse testing with the remaining 3 species, 2 Heterorhabdites species and a Sternarema species. So we isolated, reared these up, got good numbers, threw them on garden beetles in soil in cups, and see who died. Well, they did cause grub mortality, and all of them caused significant mortality compared to the water control. And of these Heterorhabdites, Bacteriophora, this second bar here was caused the highest grub mortality. Mixing that with the other Heterorhabdites had the same amount of mortality. So it appears that there is some effect from this. Now taking that to a practical level where this could actually be reared up and used as a biopesticide is an area where we haven't gone yet. We did other work along the way, a lot of work. Adrian was a very industrious grad student. I don't have time to tell you about all of it. Uh, he was interested in the genetic architecture of the populations. One of the biggest questions of this is why? Why are we all of a sudden, out of the blue, practically, finding some populations become they've been in the area a while. They've been documented in our states for quite a while. Why are they just now emerging as field crop pests? Is there something about these populations that's unique compared to other populations in other places where they aren't pests? So he started some molecular work looking to find genetic resolution of different Midwest populations. We got collaborators to send us beetles from Illinois to Delaware. We had a really good transect, but we had poor resolution. The markers just were um, not fine-tuned enough to show us resolution among populations. And upshot, we need better markers in the future. And this was a side project of Adrian's. He wanted to learn the techniques, so we didn't take it any further yet. Another question we had was, why do seed treatments work so badly? Is it something about seed treatments in sandy soils? Do we get so much water just pouring through that it washes right off? Well, long story short, we did a greenhouse experiment with different soil types and seed treatments and looked at the degree to which the product ended up in the plant. Bottom line, washing off in sand does not really seem to be the factor explaining the poor performance of these products. We did a study looking at host plant preference of grubs in the lab. If they get to pick what they're feeding on, what do they feed on? Upshot there, they really like corn. They really like corn roots, more so than soybean or mare's tail, even though they will eat those two things. And in fact, the adults loves mare's tail, but the grubs really like corn. Chris Stefanzo did a very nice experiment in Michigan looking at the relationship between stunting and yield loss. Now, when a plant is killed as a seedling, obviously that's 100% yield loss for that plant. Dead plant doesn't have any yield. But when you have these stunted plants, they do grow up, they are there in the field, they do have some corn, but how much yield are you losing? So she did a very laborious experiment where she tagged individual plants that had been stunted by a garden beetle feeding, tracked them all the way to yield, took the corn cobs, measured the various yield components, and was able to actually pin down the relationship between the stunting and the resulting yield loss. We have what I call sort of a provisional economic injury level. I wouldn't call it a threshold because there is no rescue treatment. It's not like you can say, oh, I have this many grubs, I can take action. Like, once the thing is planted, the ship has sailed. The only thing you can do, however, is replant. And so we have found that when you average in a sampling area 1 grub per cup cutter sample, you are at a meaningful economic loss situation. Much of this work is published in the scientific literature. Some papers are still in the process of being published. We have put out 3 different extension publications. We have a fact sheet on Asiatic Garden Beetle that has been updated at least once with new information. We have a very nice little pocket field guide to Asiatic Garden Beetle. This is a small spiral bound notebook. These are very popular with growers and crop consultants. We also have an EPN, nematode, fact sheet for people who are interested in this, with also some how-to on how you can isolate nematodes from your fields and rear them up for anybody who has interest. I think this would be a cool like 4-H project, so it might be nice to develop this for students in 4-H. So I could go on all day, and I have gone on all day about Asiatic Garden Beetle, but for the moment, I will leave it there. I want to thank a bunch of people, in particular Amy Rodenbush, the lab manager of my program, who none of this would happen without her. Megan Zare and many summer helpers who helped do a lot of the work. And I should say that when I say we did something, I really mean they did it. I would like to thank our funding sources, our USDA NEFA grant. Adrian got a SARE grant to help support some of the nematode work. Elson Shields and Tony Testa from Cornell, who taught us how to nematode. And several county extension educators who were extremely helpful in finding fields and willing farmers for us. And this was such a laborious project that even the baby helped now and then. So that's all I have for you for now. I am happy to take your questions, and I thank you for your attention.
Thank you very much. That was a great presentation. So yes, anyone attending, please type any questions into the Q&A portion there at the bottom of your screen. So while we're waiting for those people to type in questions, just Oh, lovely picture. Just a quick question, without kind of rescue treatments, are farmers scouting for this? Is fall scouting worthwhile, spring scouting?
So spring scouting isn't really very doable for the average person until after the planting period is over because they're so far down in the soil that you're not going to easily find them. And then they come up in that sort of April, May area time frame, which is also when the planting happens. So um, usually by the time a problem is known, the horse is already out of the barn. What you can do is scout to, once the grubs start coming up, you can scout for an early warning about, am I going to need to replant? And what parts of the field are going to, how much area requires replanting. Now, something we are trying to develop is, where this is work and it's still in progress, is fall sampling. And what can fall sampling tell you about what you're going to need to do next spring? Because the eggs are in the field in the middle of the summer, the grubs are starting to grow in the summer, fall, so they're going to be there the next year. So what can you learn in the fall that will help your management in the spring, whether that's choosing products that you can use at planting, whether that's planting a different crop, whatever it is. And that's work that's still in progress there, but we can find them in the fall, and I think that's going to be ultimately the most useful thing that people can do scouting wise.
Okay, are there, I mean, they were less preferential to soybeans. Are there other crops that people are kind of rotating in if they think there's gonna be a problem?
That's an area where we really need more information. I um, feel pretty confident there are crops out there, whether it's a type of cover crop or some other type of cash crop that would be less favorable to them. I know anecdotally from just messing around in the greenhouse that they do not like sunflowers. Now, that's not really a cash crop in Ohio, but if you really have an AGB problem, some of these guys really have an AGB problem and they're at their wits' end. Maybe it's something you could throw in the field. That's a great area for future research.
Okay. We had a question come in from a participant. Were infested fields in close proximity to neighborhoods where they may have jumped over from turf slash ornamental heavy areas?
That's a good question. We were trying to get at some of the landscape factors, like looking at the landscape and kind of big picture, what landscapes tend to have the most of these running around. Most of our fields are in heavy duty agricultural areas, and there's not a lot of, residential slash turf, in the town. There will be some turf, but not really out in the countryside. It's just not that kind of area. Uh, at this point, really, we can find the beetles in turf all over. We've looked all over. We've got it on our own research farms in the sod rows, but just not at that type of pest level. So really the biggest predictor that we have is sand. And why? Don't know yet. We think maybe it's because it's makes it a lot easier for them to move around and they just find it physically to be very amenable habitat, but that's just guesswork at this point.
So the areas where they've, I mean, have they spread much beyond the initial areas?
Well, it started out on the East Coast about 100 years ago, and as of the early part of the 20th century, they were as far west as Kansas. So they've been here.
Mhm.
We know they've been in Ohio. I think the first in our insect collections, the first specimen dates to the like early 90s. So they're here and they've been here. They just haven't been a problem. And so that's really fascinating, isn't it? Why is that? That's a
Mm.
question we haven't answered yet.
We'll have to have you back when we get
Yeah, in
done
10 years,
with
I'll probably know a little bit more.
this.
Any other chat questions for me?
Don't see any others coming in. So again, if you have any last questions, please throw them into the chat or the Q&A. In the meantime, thank you so much.
Pleasure. And let me just add that I believe the organizers have put 3 links in the chat to those 3 extension publications that I showed at the end. We have those as hard copy, but we also have them as PDF online if you want to look those up.
Yeah, those are very helpful. I like the idea of a kids project with the nematodes, a kind of a 4-H sort of youth project would be fun. So if there's, feel free to type in any questions, but let me just let everyone know that our next Pest and Progress webinar will not be until January. So you get a break until next year, January 15th, 2025. Again at 12:30 Central, 1:30 Eastern. And we're going to have epidemiology and measurement using tar spot as a model with a brief summary of tar spot inoculation experiments in semi-controlled environments. So that will be Dr. Christian Cruz from Purdue University will be giving that seminar. So please again, check out the links in the chat. Also remember, subscribe to our newsletter and see any upcoming webinars in there. So we appreciate your time today. Learned
Thank
a
you so
lot.
much for having me. Yep, it was
Love the
fun.
raccoon story. That was the best email ever. So have a great rest of your day, everyone.